Domain 8 is research transparency. Now, the purpose of this domain is to assess whether the animal study authors were transparent in their reporting. Transparent reporting increases the confidence in the study findings and promotes replicability of the research findings. Reporting guidelines such as Arrive guidelines, which is in version 2, gold standard publication checklists to improve the quality of animal studies, National Research Council's guidance on description of animal research in scientific publications can help with transparent reporting. Signaling question 8.1. Did the authors describe the experimental procedures sufficiently in a protocol that was registered prior to the start of the research? Authors may have changed the protocol of the study in various ways in addition to change in outcomes, which are covered under internal validity for in selective outcome reporting specifically. For example, the model, intervention, control, or methods of administration of the intervention and control could be changed. So the selective outcome reporting covered only selection of outcomes. Here we are talking about change in the model, intervention, control, or methods of administration. The experimental protocols should be registered prior to the start of the study in a preclinical trial registry, such as what's shown there, preclinicaltrials.eu, which allows registration of animal studies and is searchable. Studies can be registered in open science framework, open access preprint servers, print, online journals, institutional or public data repository, so for example, Zenodo. The study author should provide a link to this registered protocol in their study report. This question differs from signaling question 5.1, which refers to the description of the final protocol used after the modifications to the registered protocol in sufficient detail to allow replication. So in 5.1, we asked whether the protocol was could be replicated, sufficiently described enough to be replicated. Here we are asking, did the authors describe the experimental procedures sufficiently in a protocol that was registered prior to the start of the research? We fully acknowledge that during the process during the experiment itself, you may have to make changes in the protocol. That's absolutely fine. It's about being transparent as to what you originally started with, why you changed the protocol, and being transparent. So how would you classify this question? Classify this as yes or probably yes if there is a registered protocol that is registered prior to research commencement and that had enough details to allow replication. Classify this as no information if there is no link to a registered protocol. So you don't have to go and search for a protocol. The authors should provide a link to the protocol. So if there was no link, then no information. Or if the authors provided a link to a registered protocol, but it was not clear whether the research commenced before or after the registered protocol. So they might have registered the protocol after the experiment commenced. So they finalized all the protocol and then registered it. Or if it was not clear whether the authors provided sufficient details to allow replication. So, if it was not clear, you're not clear whether there are sufficient details to allow replication in the registered protocol, then again, you would classify this as no information. Classify this as no or probably no if the authors provided a link to a registered protocol which was clearly registered after the research commenced. Or if it was clear that the details available were clearly insufficient to allow replication. So they mentioned we did this experiment in mice and we used the protocol is described in maybe a small paragraph. which doesn't describe the details. Next signaling question 8.2 is, did the authors describe any deviations from the registered protocol? As mentioned previously, there may be justifiable reasons for alteration from a registered protocol. Authors should be explicit and describe any deviations from their plans and the reasons for them. In addition to registries, repositories, and journals for registering preclinical trials, some journals also offer registered reports publishing format, which involves peer review of the study design and methodology, and if successful, results in a conditional acceptance for publication prior to the research being undertaken. This will also allow evaluation of the deviations from the registered protocol. So how are you going to classify the signal in question? Classify this as yes or probably yes if the authors described any deviations from the registered protocol. Classify this as no information if the authors provided a link to a registered protocol, but it is not clear whether there were any deviations from the registered protocol. Classify this as no or probably no if the authors did not provide a link to a registered protocol. Signaling question 8.3, did the authors provide the individual subject data along with explanation for any numerical codes, substitutions, or abbreviations used in the data to allow other groups of researchers to analyze? Only a third of computational and data analysis could be reproduced in a straightforward way or with minor difficulty. The remaining required substantial revisions for reproduction or could not be reproduced at all. We are talking about analysis, so we already have the results. We are not talking about replicating the protocol or reproducing the results. We are actually talking about the analysis. So you have the results. Now we are analyzing the results from these tests, but only a third of the computational and data analysis could be reproduced in a straightforward way or with minor difficulty. The remaining required substantial revisions for reproduction or could not be reproduced. Availability of the raw data from which results were generated, the computer code that generated the findings, and any additional information needed such as workflows and input parameters are key aspects of reproducibility and replicability in research, in addition to a sufficiently detailed protocol. Okay, so you need to know, describe how to analyze the results as well. The data can be provided either as a supplementary appendix in the journal publication or can be provided in open access repositories such as zenodo.org or university open access repositories. So how will you classify this question? Classify this as yes or probably yes if the individual participant data with sufficient details to reanalyze were available. Classify this as no information if the individual participant data was available, but it was not clear whether the details were sufficient to reanalyze. Classify this as no or probably no if the individual participant data were not available, or if they were available, it was clear there were insufficient details to reanalyze. Note to you or the assessor: if the individual subject data is available after signing a data sharing agreement or by writing to study authors, please classify this as probably yes. Okay, so if they add a note, if you want the individual subject data, please contact us. That is fine. We are talking about people willing to share the data. Okay, so that's the end of this signaling question and the details of the tool.